No.
Instead of just writing control, they had to write con.
Con on it.
It's confusing, so it's like you think it's another group, but it's just a control.
It's a convention. I think lots of papers just write con.
Really?
For control. I think. Yeah, really? So, okay. Maybe not lots of papers. I've seen it before.
Also, I understood this paper. This is the first paper in the class I've understood perfectly until figure 4. And it's just like.
Figure 4 is what? So, okay, well, that's. It sounds like you did your job. You know, the idea is to try to maybe less than struggle, but something like struggle with it a bit, and then I figure 4 is the flux.
How they measured insoluble. Yeah.
So we'll say hi. We'll start with the question page, and then move on to the article. So lots of you weren't here when we finished the G protein couple receptor, so I hope you try to go over the material. And as I wrote in the email, if you need any help outside the class, don't hesitate. So let's go over the question page. So question 1. Okay, question 1. When signaling molecules are received by the same cell that secreted them, the signaling is called? Sorry? C. Autocrine, yes. So auto is when something acts on itself, so autocrine signaling. Question 2. Which sentence is incorrect about G proteins? So C, the trimeric protein is transmembranal, correct. It's only anchored from the cytoplasmatic side. So it's not transmembranal, but let's just to go over the material again, so A, B, and D are correct, and so the protein that regulates the proteins that regulate G proteins are influenced by outside signaling. Obviously, we mentioned GAF and GAP in this sense. The alpha subunit in the trimeric protein is homologous to the monomeric protein, what is called the RAS subfamily. And D is the alpha subunit is capable of GTP hydrolysis. Remember, a GDP is being exchanged with the GTP, which is then hydrolyzed by the G alpha. So D is also correct. Okay, 3. Below are the different steps in the signal transduction pathway of G protein via adenylin cyclase. Adenylin cyclase. That might be a mistake here. In an arbitrary order, and you need to arrange the steps in the order of occurrence. So what happened first? What happened last? What's the first step? So what? Number 3, right. So G protein activation. Then we have adenylin cyclase activation, 1, right? And then, right, 5, increasing cyclic AMP levels, proceeding with PK activation, number 2, and ending with phosphorylation of targets in the cell. So the whole sequence for this question is 3, 1, 5, 2, 4. Which is the code for the machine. No, okay. So question 4, some kinase activation leads to phosphorylation of the same receptor that causes the activation. How does this phenomenon called? C, not C, D. Homologous desensitization. So again, the kinase phosphorylates the receptor that initiated its activation. Right. So this is when when the kinase is downstream to the same component that activated and it phosphorylates it, then we call it homologous desensitization or homologous phosphorylation. Sorry? Yeah, of course. So if you have on the membrane a receptor, one kind of receptor and another kind and a different kind, and if you activated this receptor and downstream it activated a kinase, let's say kinase X, and it. So if kinase X phosphorylates the rectangle, the triangle, and the circle, we call it heterologous phosphorylation, or desensitization if it only phosphorylates the receptor that initiated this cascade. So we call it homologous desensitization. Sorry? Sorry? Only one, and it needs to be the same one that activated him. On other kinds of receptors. Sorry, again, what case are you describing? I think I didn't understand. Yeah.
One another.
Who? This kinase phosphorylated
by the
yeah. It phosphorylates only one of the other. Not that I know of. No, I don't think there's such. There's the thumb rule that you can never say in biology that there is no such thing as. Well, for these two phenomena, we have a term: heterologous desensitization and homologous desensitization. That's the only phrasing I can commit to.
So the hetero is for phosphorylating some kinds of receptors.
Different kinds, all kinds. It's a variable way of phosphorylation. Yeah. Okay, question 5. The arrestin protein. So which sentence is correct here? B, right. The arrestin protein binds to a receptor and prevents activation of a G protein. It does not bind to the G protein and does not bind to the ligand that prevents the activation and prevents the activation, because the ligand is extracellular and arrestin is cytosolic, and it also doesn't bind to the DNA because it's cytosolic and not nucleic. It is not located in the nucleus. Okay, so that was G protein coupled receptors. So let's now move on to the paper. Okay, so the title is amyloid beta protein dimers isolated directly from Alzheimer's brain impair synaptic plasticity and memory. So if we deal with Alzheimer's disease, let's start with the most general question: what is Alzheimer's disease? What do you know about Alzheimer's disease? Right, so that's the clinical manifestation of Alzheimer's disease, dementia. Well, there's a common path to almost all patients in Alzheimer's disease. Usually, patients start with forgetting or having trouble with making new memories. Then it proceeds to deficits in short-term memory, and eventually they have a very substantial loss of even long-term memory. So that's the clinical manifestation. What about the pathological manifestation? What happens at the molecular level? What do we see in the brains of Alzheimer's disease patients? Right, so accumulations, protein accumulations or protein aggregates of a peptide, a peptide that is called amyloid beta peptide. So it's the monomer. Amyloid beta peptide is the monomer for the plaque, which is the aggregated form of the amyloid beta. Where does amyloid beta, the peptide, where does it come from? Is it exogenous, endogenous, from which? Endogenous. So all of us have amyloid beta in our cells, and all of us have the protein from which amyloid beta is cleaved, which is which protein? APP, which is, it's easy to remember because it's amyloid beta precursor protein. That's what the APP stands for. So, and that also tells you that we know that APP is transmembranal. We know that it's there. We don't know its function. The only function that we attribute to this protein is that it's an APP, amyloid beta precursor protein. It has a function, most probably, because otherwise, why should it be there? But we don't know its function. It's an interesting question by itself. So amyloid beta is cleaved from the APP, which is a transmembranal protein, and it's then released to the environment, and after that, aggregated and does all sorts of things. Okay, what are these things? What are the amyloid beta induced effects? Okay, so that's the main contribution of this paper, right? It caused deficits in LTP, in long-term potentiation, and also in long-term depression. By deficits in LTD, we mean strengthening of the LTD. But before this paper, what did we know before that about amyloid beta? What do you think it caused in the disease, for instance, in the human disease, not in some experimental system? So amyloid beta, what are the induced effects besides deficits in LTP? So LTP is a toxic species molecule. It's a toxic agent. What do we mean in biology by a toxic agent? I said LTP, sorry, of course. Amyloid beta, sorry. Amyloid beta is a toxic molecule. What do you mean by toxic molecule in biology? Okay, so the most common, agreed upon everyone definition is that, and the classical definition indeed is that you add it to some experimental system, you grow cells or something, you add amyloid beta, and the cell starts to degenerate and eventually to die. But the term should have been, and indeed have been, extended to include a more subtle effects. And a toxic species would not be only a molecule that, when you add it to the system, cells start to die. So, for instance, what they show here are much more, you would agree, much more subtle effect, that you have deficits, even not in the basal synaptic transmission, because they show it in the first 20 minutes of recording. We will get to it in a minute. But only when you try to induce LTP, this is when you see the manifestation of the amyloid beta. We don't talk about toxicity in the sense of dying cells or anything. And what other? There's one more thing that people have shown before this paper for amyloid beta, and that is that the spines, the dendritic spines, the structures, the postsynaptic structures, will change the structure and their morphology. We do not necessarily, before this paper, could have said that this is a damaged effect, a damaging effect, but indeed there was some effect on morphology of dendritic spines. And this paper really pushes the field a lot towards what are, and then lots of other research groups did very similar experiments. What are the readouts that we need to check in order to talk about toxicity in the context of Alzheimer's disease? Because, and by the way, just last comment about this issue is that, well, if a neuron would have some synaptic deficits, either in LTP or basal transmission, then it eventually will degenerate and die. Maybe after a couple of years, but eventually it will cause its death. A neuron has to have some basal level of functioning to be healthy and to be maintained properly and for a long duration. And so that's also toxic, but just for the far future, perhaps. Okay, what are the soluble and the insoluble forms of amyloid beta? We already actually mentioned it. But in this paper, they call it the soluble form and the insoluble form. Which is which? What are these two? Sorry? Yeah, so okay, soluble massis and insoluble kshetemes, I'm not sure. Okay, so the soluble is when the A-beta is cleaved from the APP. It's a peptide, it's soluble, it swims in the cytosol or in the exoplasm space, wherever, and then it starts to aggregate and becomes insoluble. So you can wash the soluble, for instance, form and leave and be and left with the insoluble form in some test tube or colon or wherever. Okay, so that's Alzheimer's disease in a nutshell, and a word about amyloid beta. Okay, let's start with figure 1 from the paper. So what are these gels represent? What did they run on these gels? What is the technique here?
Western blotting.
Sorry? I didn't hear.
The Western blotting.
Again, sorry, the what one?
Western blotting.
Western blotting, yeah. So that's Western blotting, right? And before Western blot, what is the preceding step? Right, so that's a technique we discussed in one of our classes. So what do you do before a Western blot? The blot is when you take the something that I'm asking you about, and then you blot it, you transfer it to the membrane, to a membrane. And then you mark it with an antibody, the membrane. But before that, you had to what makes this ladder here? What makes these proteins run or be located in different positions on the gel according to their size? So no, it's not immunoprecipitation, which is the technique they use to isolate the amyloid beta from the patient's brain. No, so I'm asking you about, I think you're right, but I'm asking about the technique itself. How did we call the technique in which you separate proteins on a gel by their size? Gel electrophoresis. So if you said it before, I didn't hear it. Sorry. So it's gel electrophoresis. That's the first step. That again, that separates proteins by their size, and then you blot it to the membrane. And then we continue with you. Okay, so after you have the membrane, what gives you the dark signal here? Why do we see black here and white here? I mean, when you take the membrane, you don't see anything. So we had to stain the membrane, right? And we do it with all the procedure we mentioned before of a first antibody, then a second antibody with an enzyme in its edge, and then you pour some substance on all the membrane, and only where you had the enzyme that transforms the substance to a makes it fluorescent, then you get a signal, and then you can measure or even quantify the level of intensity of these dark signals. Okay, so that was about the techniques. What are the three different gels for? What differentiate between each one? The what? So this is a lane. Each column on a gel is a lane, and the brain regions are separated. I mean, each brain region is in its different lanes. And FT here are for what? Frontal lobe and temporal. And then each pair is right, the patient and the diagnosis of the patient. So there are several diseases that are being checked here. This is the first 80 patients, the second and the third. And you also have, for instance, frontal lobe dementia. What's FTD? Frontal temporal dementia. Okay, so different kinds of dementia. Is it in plural? Different kinds of dementia diseases, and only AD, or the most convincing picture is for AD, in which you see the monomers of the amyloid beta, and you can, by size, distinguish between the monomer, the dimer, and the trimer. So all of these together are called low-N oligomers. Because they're oligomers, remember, oligomers are just a few mers, and they are low, so you can talk about an N oligomer, like a 7 oligomer, 8 oligomer, low N oligomer, meaning somewhere between monomer and, I don't know, 10 mer. Low N oligomer. Low N, just the letter N. So that's how they call it in the paper, so I just wanted to mention it. They call it low N oligomer. Okay, so again, I'm coming back to the question I asked in the beginning. So what is the first gel, the second, and the third? Never mind which is which, but what differentiates between the three gels? So soluble and insoluble. So again, each one, I mean, the insoluble actually, right? The plaques themselves did not go into the gel because they have a huge size, even above 188 kilodalton. So they stay somewhere here and they cut it off from the gel. And then you start to see the peptides that migrate north, to the south, and then you see the monomer. So it's not a soluble. In each gel, you have the soluble form and the insoluble, but I took it off. The three gels are different preparations, different kinds of extractions from the patient's brains. So each time they took a piece of brain, and then they can extract, and it makes a lot of difference how you extract your protein, the whole protein population from the tissue. So for once they use formic acid, and then they use SDS, which is a detergent. We will not go into the detail here, but let's just say they did a lot of work to be able to show a pure protein extracted from a human patient. Usually what we do is extract amyloid beta from cells we grow on a petri dish, which is a much cleaner system, you would agree. or you can do it from the brain of mice, but this is from patients. And patients have postmortem delay, they have different genotypes, they come from different populations, they have spent a lot of time in the brain bank, usually in the Netherlands, and eventually to be able to extract a protein that looks very good on a gel, it's a hard work. And so they tried a lot of different techniques, and they ended with some protocol, and never mind the details. Okay, so again, just note that the Alzheimer's disease patients are the ones that, from where you can extract the most significant amount of the low N oligomers of amyloid beta. Okay, so you see it in all three preparations actually. Some less, some more. Never mind. Okay.
So what are the differences
between the preparations? So in one preparation you use a very strong acid. In another one you use a very strong detergent. Never mind the details. Let's go on. I'm just.
So why do they say here that like A is insoluble and C is insoluble?
In C they say that they didn't use any technique. It might be. So maybe only the first couple. Okay, you might be right. And check for a minute. So C. This is C. But it can be. We see the oligomers. Wait a second. So insoluble C, GUHCl, sequential extracts of frontal and temporal, blah blah. But okay. No, okay. So I didn't even mention the immunoprecipitation, and that was in purpose. But then, okay, so the Western blot analysis was performed on supernatant of the soluble for every gel. So the supernatant is that after centrifugation you have the sediment and the supernatant at archiv. So you take only the supernatant. That's the one you run on the gel. I have to read it more carefully. Let's go on. It might be a good question, but let's go on anyway. Why did I put it right in the middle? Let me move it just for 1 second. The animation was gone. That's the problem. Okay. So let's start with this panel right here. So what are these graphs, which are all over the paper? What do they represent? Sorry? Right, LTP or LTD, that's the most final endpoint of this graph. But let's go a few steps backward. What is the experimental system here?
recording from a cell in culture?
They record from
A cell in culture?
a cell and?
In culture? Or
In culture. So it's not culture, it's not, it's a slice, hippocampal slice from mice. Right. So this is not from humans because you have to have it in a viable form. So you have to take it from a living mice right away, and then the mouse obviously it dies, but then you proceed with the slice. Okay, so this is what we call acute hippocampal slices. They are about 300 or 250 micrometer in their thick, and then you can And then where did they place the electrodes? Anyone try to go into these details? So you have a stimulating electrode, and you have a recording electrode. The stimulating electrode sits on the shuffle collaterals. These are the projections of the neurons that sit in the CA3 region of the hippocampus. You have the basic anatomy of hippocampus. So I'm not going to draw the whole hippocampus, but just so if these are cells in the this is the soma, right? The cell body that sits in CA3, and it sends an axon, so it has a dendrites, and someone does another neuron outside this figure perform a synapse on its dendrite, and it sends an axon and perform and have a synapse on the dendritic tree, very ugly dendritic tree of cells that sits in CA1, and then they send an axon to someplace else. The whole signal direction in the hippocampus comes from the entorhinal cortex, from some region that sits around here, and does something on CA3. This is 3. and then the signal proceeds to CA1 and move on. So you have to, so the electrode, the stimulating electrode, sits here on the axon of the neurons from CA3. We call this region the shuffle collaterals, because you have not only one, but actually a lot of parallel fibers right here. All of them do synapses right here. And then the recording electrode is on the dendritic tree of the neurons that sit on CA1. So this is recording. And if it was only one signal, what we would have seen if we will stimulate right here is a very tiny excitatory postsynaptic potential, right? Excitatory postsynaptic potential is the signal that you record from the dendritic tree at the postsynaptic site. But the case here, it's a very special case in this region of the hippocampus that you have lots of parallel fibers that perform the same synapse on right about the same area. So this synchrony gives you the ability to record not just the signal EPSP, but a population EPSP, which is here called the F EPSP, which is field EPSP. So you get some kind of like a giant EPSP, and that's what you see in the insets there, the traces, right here. This is a giant EPSP recorded from the recording electrode. And it's just due to, so this is why people use the hippocampus CA3, CA1 network all the time, because you can, it's a relatively easy recording technique. So this is okay, these are where these traces come from. And what is the y-axis here? But of what parameter? Before or after, right? But of what parameter? What did they measure from the EPSP? The what? What do you mean the potential? The voltage itself? No. Because each slice, due to, I don't know, the weather or the mouse even, has different absolute potential. But what we look at here is the difference between before and after, as you said. So just read the brackets, right? It's % of baseline slope. So the baseline is what happened, right? But the slope is the main idea here. So each trace you can model it, or you can extract certain parameters from it. You can talk about the amplitude, right? How low, not the absolute, but the delta, how low this minima is. You can talk about the slope of the falling phase. You can even talk about the slope of the rising phase. And the most common thing is to calculate the slope of the falling phase, this phase, and that's because it has the best correlation with postsynaptic change in conductance. Never mind, but that's why people usually use the slope from these traces. And here you see % of the baseline slope. So each dot here had a number, -1, -1.2, -1 point whatever, and then you divide each of these numbers by the mean of the numbers of the slopes before you did the LTP induction. That's the baseline. So this is the y-axis. And about the x-axis now. This is in minutes. Not milliseconds, not even seconds, that's minutes. So you see that after the LTP induction, we would proceed with the recording for a full hour. And then this summary is actually where did we stand in the last 10 minutes, the mean around the last 10 minutes of the hour, and compare with the 10 minutes from the baseline window. So you see that most of the time, or the black and blue, which are the different kind of controls, we sit around somewhere between 100 and 200. So this is 1.5 increase or fold change in the slope. How did they induce LTP here? Let's start with Kandel, the paper we read a couple of weeks ago. He also performed LTP experiments using a chemical induction. He added serotonin. That's also called an LTP. But here they use a different technique, which is what? Stimulation. Electrical stimulation. A high-frequency electrical stimulation. And you have to add the high frequency, because, okay, so the x-axis here, each dot here is being attained, every 20 seconds, you stimulate the cell only with a single pulse, and you monitor how steep the slope was here, or how low was the amplitude, or something like that. So you do this paired stimulation and monitoring every 20 seconds for a full, more than an hour, for about 80 minutes. And then in a certain point, you don't inject only a single electrical pulse, but actually a couple of hundreds of them in a very high proximity to one another. It's about, usually people use about 100 hertz stimulation or even 200 hertz stimulation for a second or for 2 seconds or for a second, wait 20 seconds and then another second. There are different protocols for doing this, but they didn't use any chemical agent here. They just performed high frequency stimulation, which gives you a very robust, as you see, long-term potentiation. Well, this is the short-term potentiation, but then it even lasts for after an hour about 150% from the baseline. Actually, it does. Again, so the control, the AD, the LTD, the depression, which is, again, not high-frequency stimulation, but low-frequency stimulation right here. The short term, sorry. Yeah, the short term, well, yes, you can say that. That the short term, I don't see any significant difference between, sorry, you're correct. And LTD shows the opposite effect, so the LTD is washed quickly in the control, and LTP is washed quickly in AD. We like LTP and we don't like LTD, so as a whole, Alzheimer's is bad. But the main thing that is argued here, and it's a novel thing compared to what we knew before this paper, is that the amyloid peptide itself, the soluble form, you add it to your experimental system, electrophysiology experimental system, and you see deficits in LTP. And that's, as I said, that's the summary of different experiments they did here with different extractions from different patients, and everything looks good. What is this readout? So that was electrophysiology, synapse functioning. And what is this figure here, this graph? So it's spine density. How do we measure spine density? How do we calculate? So, so the experimental system here is organotypic slices. You just grow, you can grow a slice, you can grow neurons, never mind, but you add at a certain point in their life the amyloid beta, and then you see some morphological changes. Then you can, if your neurons are nice and big enough, you can even look at the neuron with a good microscope and see spines, and you can measure how many spines you see for a single dendrite. And that's the spine density, right? How many chairs for 1 meter, chairs for a meter, never mind, and how many spines for a micrometer of a neurite, in this case, of a dendrite. Okay, so spine density decreases when you add the extracted amyloid beta from different patients. Okay, I will just put the last figure in and we'll proceed. Yes? Well, it's the same amyloid beta as a whole, but amyloid beta, even the soluble form, and this is something the paper does not go into at all, and you see some hints of it here and there in some of the figures. The soluble form of amyloid beta is classified in itself for, and it has different species of soluble amyloid beta. The one that has 40 amino acids in it, and another kind that have 42 amino acids in it. There are different kinds of different sequences of amino acids sometimes, but they all distinguish that this paper does is between the soluble and the insoluble form. But between patients, you will see different ratios of different species, or between different species of the soluble amyloid beta. But this is something the paper does not go into. What, the different ratios? Yes. So healthy individuals usually have not that few 40 amyloid, soluble amyloid beta, which is the short amyloid beta. They have a higher ratio of the 42 to 40 amyloid beta ratio. But this is a work that was done after this paper. So let's ignore it for now. and go and okay. So you see that okay. So you have two readouts or two levels of readouts here. You have synaptic functioning and synaptic morphology, right? And amyloid beta extracted from patients damaged these both of these readouts. Okay, this paper was written in 2008, and then there was a big trend to try to passively immunize against Alzheimer's disease. Passive immunization is when you inject a patient with ready-made antibodies, in opposed to active immunization when you inject some weakened form of the toxic agent of a virus of a bacteria, and then the body needs to produce its own antibodies. So that's active immunization. You just synthesize proteins, somewhere usually in the Technion in Haifa, and then you inject these antibodies to your patient. The antibodies that you want, that you think that are protective against the toxic species. Okay, so so there was indeed a lot of experiments, there were a lot of clues about passive immunization before this paper, and I think that this paper pushed this field towards the clinical trials, which I will say a word about them in a minute. But the good thing that this paper shows about the passive immunization is when you add, so that's the ADTBST is the passive immunization. So when you add only, the blue is when you add only the amyloid beta, and then that's when you have, sorry, the red is when you have, is when you add only the amyloid beta, and then you see a quick washout of the LTP. So that's why the patient forgets so quickly. And then when you passively immunize against amyloid beta, adding together the amyloid beta, obviously, so you mimic, like, you sort of mimic in your experimental system the disease together with passive immunization. So you restore perfectly, actually, the level of LTP, or the strength of LTP, an hour after the induction. So as I said, and then you have also rescue at the morphological level. Okay, so things looked good, and indeed clinical trials were initiated, and even there were some hints for good things happening. But after I think a couple of months, I think 2 or 3 patients from a cohort of about 8 or 10, so that's a lot, developed acute inflammation in their brain, what is called encephalitis, like water in your brain. That's what encephalitis means. And the clinical trials were stopped, obviously. And I'm not sure where the field of passive immunization in Alzheimer's disease stands today, but it was stopped just due to some to bad clinical trials. But the idea or the concept, I think most of the researchers will tell you that it still holds. Okay, we will skip this one and end with the last figure that says the most definite thing you can say about the difference between the plaque, meaning the insoluble form of amyloid beta, and the soluble form. And before that, so just a word about the plaque morphology. So each amyloid beta protein aggregate, is built of a core, meaning a very dense region in the middle of the plaque, and around it a more sparse or a more diffuse region, which is still part of the plaque, but it's much less concentrated, much less dense. All this plaque, the green and the blue, is built of the monomer amyloid beta. But the difference is that the plaque is not in a chemical steady state with the environment, in the sense that amyloid beta does not taking out from the green and taking back and get in back again. And from the diffused region of the plaque, the blue one, it is in a chemical equilibrium with the environment. So these are soluble amyloid beta, and they go into the diffused part and go out again, and go in and go out. And then more matured, that have been modified chemically in different manners, that lots of them we still don't understand, it becomes the core of the plaque. Okay, so what they do now is that they clean the plaque and remove all the blue and stay with the core. You do it by sequential SDAs washes. You just wash your hands very, very good. 1 time, 2nd time, 3rd time, very obsessively, and then after the 4th time, you don't see any. You still have this in your system, but you've succeeded to wash the blue part um, לגמרי, totally. Okay. And okay, so this last piece of gel here is to show that if you take the core itself and you do an FA formic acid treatment on it, you can extract soluble plaques, amyloid beta peptides from it, just to show that the toxic agent is still here, but it was just chemically modified to be very stable and not with a chemical equilibrium with the environment. Okay, so these are nice pictures of the core of the plaque. And now, if you add only the core to the electrophysiology experiments, you do not see any LTP deficits. So it's not a toxic species at the level of the synapse, right? If you add the FA, not the FA, but if you take the core and then dissociate it with formic acid, you again have the LTP deficits. So that was the last nail in the coffin that says that the toxic species, the one that we need to, to go after or to try to target therapeutically, is the soluble form. And we don't need to care too much about the core, about the plaque itself. Because up till 2008 or somewhere around there, the first pathology that was seen in Alzheimer's patients by the Alois, I think he was called, an Austrian or a German physician, I'm not sure. So he saw the plaques. So for so many years, almost more than a decade, for lots of decades, so people try to target the plaques, to clean the brain from the plaques, but that's not, at least not the way the disease initiates. And this paper proves it as definite as you can. That's my opinion, at least. Okay. Okay, that's about the paper. Any questions? Yes.
I'm just curious, why they didn't talk at all about the tau tangle thing, the theory?
No, because okay. So extracellularly, you find amyloid plaques, and you find the peptide, the amyloid beta peptide. Inside the cells, in Alzheimer's disease, you find hyperphosphorylated tau molecules. They are toxic also. We think that they cause deficits at the I think general cytoskeleton arrangement, which will obviously be very damaging to neurons. And they didn't go into the tau at all in this paper. And I'm not sure about immunization against tau. I don't think you can really. I don't know about anything about that actually. Okay, so let's take 15 minutes break and we'll proceed with the chapter.
How the plaque starts. How it
It's almost not at all. If any, so there's one thing.
why the
No, you know what? No. And that's, by the way, part of the reason. It's innate nature medicine. The people that care about results and not mechanism. I mean, physicians. So, doctors, physicians, clinicians. Anyway, so indeed, these data almost only show the endpoint of how you can, but still, they extract a little bit from human patients, and that's the first one to do it. And still, I think one of the only groups that are able to do it is Dominic. Dennis, not Dennis? Yeah, Dominic and Walsh. I met this guy. So
you said that was the hardest part, is getting that extract.
Well, everything was hard. Getting such beautiful patterns of
LTP is very
LTP.
easy though, is it, with the other side,
Yeah, it's easier up to the point you try to do it yourself, and then you find it's not very easy because I don't know, electrophysiology systems are voodoo systems. One day they work, the other day they don't.
You did the last research methods course, right?
Yeah.
Do you do
slide is and you build recipe, usually you have an open system of circulation of artificial CSS. So the CSS washes the slice continuously, and then the slice is viable and it's happy, and you get good readouts. But if you add something, and that's a very expensive something, you extract as the protocol, so you close your system and use about a 10 to 20 milliliter closed system of circulation, because you have to have a certain concentration of amyloid beta in your system, and you can't wash it out to the garbage continuously.
Right.
Because it's not an expensive in money sense, but it's ethically. People donated their brain to the science, and you have to use it very carefully. And getting a good readout in a closed system of LTP, that's hard.
Yeah.
So you have to play with lots of parameters
I didn't think of it.
to get a good readout.
בביולוגיה.
It's not easy anyway. Yeah, it's good. Nature medicine, it's pretty serious.
הבנתי. טוב, זה קונטקסט דיפנדנט. שאחד מהם אתה יכול לפתור אותו בגלל זה, לא?
זה לא כולם.
לא בהכרח כולם, כן. אוקיי. זה עדיין קל באופן מחשיב, אבל...
זה לא קל באופן מחשיב.
לא, מה, צריך להוכיח את זה כאילו?
כן, להראות את זה.
את איזה הגדרה פשוט, אני חושב, לא? כאילו, סעיף A, שאתה אומר שאחד מהם אתה יכול לפתור, זה הגדרה של דיפנדנס, מה יש פה להוכיח? זה ההגדרה.
לא, ההגדרה היא שקיים סט גלרים שלפחות אחד מהם שונה מאפס, שהוא כישרון המשוואה הומוגנית.
אוקיי, אז אתה מציב אותם כאילו, אתה מציב אותו בצד השני של המשוואה, ואתה אומר, הנה, שקלתי ליצור את זה.
לא...
ואת השני, זה נתן את זה?
מה שאלת?
מה?
למה מבחן זה?
לא.
זה?
כן. אני לא אוכל, לא יודע, המבחן זה היה לי מבחן קורסות חודש בקושי. לא עובדים שעובר הזמן. תגיד, שחר, איך זה? כאילו, אני מסתכל על המצגות של הקורס הזה, זה נראה פשוט באגן אחד גדול, כי זה, יודעים אני מעניין דברים, ולא נתחילים כאילו, לא יודע. יש לך איזה טיפים ללמידה?
לעבוד עם המקבצי שאלות.
יש מקבצי שאלות באתר?
מה שאני כל פעם שולח ויש אותם באתר. יש אותם באתר. וגם אני שולח, כל פעם שאנחנו עונים, אז אני שולח שבוע לפני זה, באימייל. אבל אני גם יש אותם באתר, ב-PDFים כאלו. וגם נעשה איזה מקבץ מיוחד כזה, נגיד עוד שבוע ניתן, נראה שזה יותר קרוב. שנסיים עוד כמעט את כל החורף.
ב-20 בפברואר.
זה קרה יותר מאוחר. מתי יש לך מבחן?
בשיעור האחרון. טוב, אני אקח את הפלאפון.
לזהות את הסדר, כאילו.
טוב, כן או הרי?
כן.
לא זה משהו.
זה אמור להיות מבחן יחסית קל, לא? טוב.
כן, אתה יודע, זה כאילו בין בוחן כאילו, כן.
מה זה? כאילו רק שאלות אמריקאיות כאלה?
כן, זאת ה-30.
עושים שאלות אמריקאיות וכמה אחוזים נמצאים בזה?
40.
40? אז מה היה 60?
אז זה כתוב באתר, תראה, יש 40% זה הבוחן, זה המבחן, 40% זה עבודה. אז עבודה שכתוב.
אוקיי.
עבודה לסיום, זה עמוד אחד של עבודה, ואז עוד 20%
תלות בשיעור.
כן,
לא עשיתי כלום. למה בעייתי?
לא, לדעתי הוא קל באופן מחשיבי, אבל לא הבנתי, כמו שקראתי פה מה זה ברק. כאילו, כל השכון פוסט מישן השבוע עליתי
כן, תרביתי איתכם, אני גם צריך.
למה?
כי ביקשתי שהוא יעביר שבוע שעבר לשבוע הזה.
אז בוא תחליט איתי, וזהו.
מה?
כאילו...
איך אני אחליט איתך?
לא, תעביר למקום אחד, כי אין לי כוח לזה עכשיו
כאילו, כבר ביקשתי
סוף את הכיתה עכשיו, אני מלמד פשוט מ-9, תודה, אני אעשה את זה בכיתה שנייה.
אם זה בסדר,
כן, אתה צריך לשאול את השקפים.
גם שבוע שעבר היינו בקטנה, ואז זה גם לא מצטלם, אז...
כן, 100%.
הנה, אחלה, תודה. תודה רבה. So we started last time the chapter about post-transcriptional gene regulation. We mentioned a couple of mechanisms that happen in the nucleus, among them splicing, and we've mentioned that splicing gives you the ability to produce different RNA variants from a single gene, which then give rise to different protein isoforms from a single gene and different functions for each one. We've mentioned the notion of consensus sequences along the introns, and we have described the splicing reaction, that is two sequential transesterification between certain regions at the exon-intron-exon region, and then you are left with two joined or rejoined exons, and with an excised lariate intron, which is being degraded in the nucleus. And we'll now proceed with the small nuclear RNA molecules that are the RNA component of the spliceosome, which is the big machine that includes RNA molecules and proteins. So the RNA component of the spliceosome is built from 5 small nuclear RNAs that base pair with the pre-messenger RNA. Before it was being spliced, we call it the pre-messenger RNA in different regions. U1, each small nuclear RNA is termed U with the number, so U1, never mind these details. U1 base pair with the consensus sequence from the 5' end, and U2 base pair with the region around the adenosine, which is the branching point. And note that it has a perfect complementarity with this region, except with the A. So the A protrudes to the environment, protrudes outside, and that's the reason why it is exposed to perform the first transesterification reaction, the first chemical attack that happens in the splicing reaction. So so this is not accidental. That's a very important feature in the interaction U2 undergoes with the messenger RNA. And these are the additional 3 U's. So the splicing reaction, as was described here, happens in cells something like this. So this is the core of the spliceosome. The core is built of the small nuclear RNAs, and they change their configuration in relation to one another in a way that each time brings the attacker to the right orientation in relation to the molecule that's being attacked, and then allows or promotes substantially the transesterification reactions. And this RNA core is not just like this, but it is actually roped with additional about 70 proteins. So all in all, the spliceosome is roughly the size of the small ribosomal subunit, which is quite big. And the proteins, again, they participate in the configurational changes, series of changes that happen along these transesterification reactions. Okay, so, and we will not go into detail in this next notion, but it was seen, and lots of evidence are being accumulated these days, that the elongation step of transcription that happens in the nucleus is coupled with the presence of RNA processing factors, such as the small nuclear RNA and different proteins that are part of the spliceosome. So again, transcription is coupled with splicing in many different mechanisms that some of them we already have some details. And just keep in mind that if something goes very wrong with transcription, then splicing will be stopped, and vice versa. There is some checkout points between these two mechanisms. Yes?
Do you know that if there is a frame shift mutation or something like that, if this frame gets too long, can splicing repair this problem?
I don't know of any specific examples of splicing repair of a frame shift mutation, but some mutations, I mean, for a lot of, ever since we have so much high throughput data about splicing, we've got to learn that lots of the mutations damage at the splicing level or at the splicing mechanism. I don't know about a repair. Might be an example of such a case. I don't know. Yeah, I really don't know. Okay, so when we talked about the consensus sequences along the introns, you remember that they were very degenerate, just very short, just 2 nucleotides in each side. So for most of genes, they are not enough to definitely or accurately define the exon-intron junctions. And so we have also, on the exons this time, consensus sequences that are recognized by proteins that are called SR proteins. Never mind what the SR stands for. Everyone has forgot this altogether by now. They are called SR proteins, and they are required to define accurately the exon that should be spliced together. So SR proteins cooperatively bind to one another and interact with with some of the components of the spliceosome, so they recruit or help or promote the spliceosome components to arrive to their correct location, and for the spliceosome to be built in its correct location. So just note that what we call a spliceosome is distributed along an intron in its 5' end and 3' end. So that's a spliceosome right here. And upon the exons, we see the SR proteins. And that's about how things look when on the pre-mRNA before it is being spliced.
What do the Ys mean on the RNA?
These Ys. No, it's not an amino acid. We're looking on a messenger RNA. We're looking at the nucleotides, right? So this something. There is some definitions about. I don't know what the Y's mean to begin with, but I don't know what the Y means. Okay. One more comment about the spliceosome. So. So here we looked at specifically on introns, right? And we said that, well, in mammalian cells at least, and actually in most eukaryotic cells, the way that this splicing reaction happens in the cell is by the promotion that happens with the small nuclear RNAs and the proteins that build together the spliceosome. But for some introns that are present mainly in certain kinds of mushrooms, so they have the ability to self-splice themselves. They have the ability of self-splicing. Now take that together with the fact that they structurally resemble these small nuclear RNAs, and you will reach the assumption that these these introns that we see in mushrooms during the evolution, preceded the small nuclear RNAs, the mammalian form of small nuclear RNAs we see today in animals, preceded them in evolution and gave rise to these genes. Okay, so these introns, you take them and you just put them in vitro in the test tube, and they can splice themselves out of the messenger RNA sequence. That's what we see. You don't have to have any of the spliceosomal components for them to be spliced. Yes?
What's the difference between the intron and the spliceosome? It's the same area?
No. So the spliceosome is the RNA components, not the messenger RNA, but the small nuclear RNA, the U1, U2, additional U's we saw in the previous slide, together with proteins. You don't see most of them in this figure here, just two examples. And this machinery, this complex here is called the spliceosome. And the intron is the area on the messenger RNA that is being spliced. It's the gray line here. So these two components are just part of the spliceosome, and this is the messenger RNA. And these are the two exons that will be rejoined together after the removal of this intron. Okay, let's see some examples of the biological relevance of splicing in neuronal systems. And we'll begin with stress, mental stress. So PTSD is post-traumatic stress disorder. So some patients that undergo severe trauma can develop chronic stress. They do not go over, recover this trauma. Some of them never, and they have a certain level of chronic inflammation, chronic stress. Okay, so we know that in these patients you find, as I said, you find high levels of chronic inflammation in the form of different cytokines and different components of the immune system that are activated chronically during their whole life, and not just as a response to some external agent. Okay. And we also know, so the stressed brain causes in the periphery inflammation, continuous inflammation. And we also know that activated macrophages, they secrete cytokines. Again, just never mind what are these molecules, but they are just messenger molecules, or sort of messenger molecules that perform lots of things. Among them, they can pass the blood-brain barrier, the BBB, enter a certain region in the hypothalamus where they will be recognized by receptors that will cause, that's what was seen a couple of years ago, that will cause a strengthened consolidation of stressful memories. So inflammation in the periphery also stresses even further the mental stress that resides in our brain. And so you have a loop in post-traumatic stress disorder. That's the most common model of this syndrome, of something that feeds itself. And okay, so this is one part of the story. And the second part of the story starts with acetylcholine esterase. We've mentioned it. This is the gene that codes for a protein that sits in the synaptic cleft, and there it hydrolyzes acetylcholine, the neurotransmitter acetylcholine, and stops the signal transmission. ACH is not only in the muscle, but it's the only neurotransmitter acetylcholine in peripheral muscles. But you have ACH in the brain in lots of areas. Okay, so there's another splicing variant of ACH. The common one, the one I just mentioned, is called the synaptic ACH, and there's another one that has very low levels, basal levels. It's called the read-through ACH because it includes some, well, it's for tag, it includes some other portion that here is really an intron. It's removed, and here it stays, and we see it in the final product, of the messenger RNA, and also in the protein itself. So we call it the read-through. And well, we don't have a specific mechanism for this yet, but some evidence show that rising level of the read-through acetylcholine esterase has to do with this cycle of inflammation between the central and the peripheral neuronal nervous systems. And it was even shown in mice that to reduce, okay, so in PTSD, there are higher levels of this read-through ACH. In most of us here, the levels of this species is very low, but in PTSD patients, it rises very substantially. And it was shown that blocking the rise of this species, of this variant, is enough in some mice models to reduce this syndrome, to reduce the stress status of the animals. We don't have a specific mechanism for how it does so. We have some ideas. We know that acetylcholine plays a role in activation of macrophages, and through them, it's a pro-inflammatory molecule, and so on. But let's keep it as simplified as I've just described. Yes. Okay, that's a good question. That's the main question for retrospective studies and compared with prospective studies, where you have a bunch of PTSD patients and you don't know if they were maybe more stressed before the trauma and so on, and you can't even know if it is a genetic component that have increased their read-through ACH, which then caused the PTSD cycle, vicious cycle, or was it just purely environmental event that caused the increasing levels of ACH. I don't think there are still studies that show this unambiguously, but at least of one study that is a prospective that takes about 200 children, and we check them year by year and hope that some of them will develop PTSD. Quite surely they would, because this study is performed in Gaza and in Sderot, so we have a good chance of PTSD there. So, and you might be able to answer more accurately these questions. I don't think anyone has any absolute answers here. Yes. Okay, so that's a portion that up till we've seen it in this messenger RNA, we've called it that's an intron actually, but because we see it in well, it's something between an intron and an intron that is being retained in the messenger RNA, so let's call it an exon. It's a portion that we have in some RNA variants, and we don't have it. It was removed in others. Okay.
What does the ACH do to the acetylcholine?
It hydrolyzes acetylcholine, and thereby causing termination of the signaling in cholinergic synapses. Because when the acetylcholine there is hydrolyzed, it is no longer being recognized by the receptors for acetylcholine, the nicotinic and muscarinic receptors, and then the signal terminates. Yes?
So, I just to make sure that the splicing is where the read-through acetylchlorine is formed instead of the synaptic one?
It's not instead, well, in all of us, you have both of these molecules in different ratios. This one is pretty low, while this one is very high. It's been expressed very high in cells. But in PTSD, the ratio between these two changes in favor of this one, of this variant. Okay. These examples, I mean, the details here, because they are not known, and you will not be asked about them in the exam. I hate to say this sentence, but just try to, at least in the examples for the neuronal system that has to do with splicing or with other mechanisms, try to sit back in your chairs and enjoy the story more than try to capture details. I didn't want to say it up till now, but just for us to be able to finish the whole chapter, hopefully today. Okay, any critical questions? Okay. Yes? Sorry? Okay. That's the difference between these two is splicing. Here you don't have the 4 point because it was spliced out. That's splicing. I mean, this molecule spliced to give rise to these different two molecules. Okay.
So
yes.
The splicing?
Sorry? Obviously something, yes, something changes in the splicing status, at least for this gene. Okay. So this it's another story, and this time the story is from the retina of Drosophila. Okay, let's start with the third point, actually. So we know that different neurons in the retina of all organisms, but specifically of Drosophila here, it's the fruit fly. Yes, for those who don't know, Drosophila is the fruit fly. It's a model, it's an animal model for lots of different systems. Okay, so this is an adhesive protein. It's a gene that codes for an adhesive protein. Adhesive proteins are proteins that cause recognition and linkage between two cells and allow them to bind together or to stay locally in close proximity to one another. And we know that these proteins play a part in the way that retinal neurons interact with lots of different brain areas. So the retina, again, interacts or sends projections to lots of different brain areas. And we think that it does so by the function of different adhesive proteins, among them DSCAM. Okay, so there's a small maybe subfield in science, in neuron science, that try to explain how do we have so many different interactions, how does a neuron know to send a projection to this neuron, not to another, to this brain area, not to another. And here you have to explain in a very relatively sophisticated manner, because again, retinal neurons, in opposed to other neurons or more than other neurons, send projections to many, many brain regions. Okay, so take this together with the fact that the DSCAM in Drosophila is a huge gene. It has putatively around 95 exons that can be alternatively spliced. So 95, if you do the combinational math, you get about 40,000 possible isoforms for this specific gene. Some experiments also show that indeed during development you have some the splicing status of this gene changes. And so this could give us a mechanism through which neuron X knows to interact, to send a projection, recognize and adhere to this neuron and to that neuron, to the neuron in the hypothalamus, in the VA1, and so on. Okay, so this, and we don't have a lot of details about this one. I just describe this sort of subfield in neuroscience. Okay, this piece of work is in a much more mature state, and we know a lot about it already. Okay, so this time the system is the auditory system, which you will hear a lot about with Elin Elkin in physiology, but he would not mention the fact that splicing also helps the tuning, though the fine tuning, but still the tuning of hair cells in the cochlea. Okay, so hair cells are the cells that transmit sound waves to neuronal activity. They sit on the basal membrane, or that is in the cochlea. Okay, so about the general anatomy here again, you will learn this with Ellie. But what we do know is the different hair cells, not only due to the fact that they sit on different locations on the cochlea. Okay, so different things cause hair cells to be sensitive to one wavelength, to one sound wave and not to another, because of different reasons. Some of these reasons, one of these reasons is because they sit at different positions on the basal membrane, and the basal membrane in its turn is mechanically sensitive to high frequency in low frequency in one region, and to high frequencies at another region. So that's the first determinant to which sound wave the hair cell will respond to. And there are others, anatomical aspects of this thing and so on. But also, different hair cells are tuned to different frequencies due to intrinsic oscillations. Even before the arrival of any wave, sound wave, these cells continuously oscillate. And they oscillate in different frequencies that are determined by a potassium channel that opens in different thresholds of calcium concentrations. Okay, so it's the calcium concentration changes, never mind now why, but that's what causes these channels to open and close and causes the hair cells to oscillate in different frequencies. Now, the gene that codes for this potassium channel is called slow, and you have lots of different splicing variants for slow, and it opens at different calcium concentrations. So each variant of slow would cause the hair cell to oscillate in different frequency, which will cause the hair cell to be finely tuned to specific sound waves. Okay. And there are at least 8 known splice sites, functional splice sites for this potassium channel, the slow, which again, as in the DSCAM, you have to show it all the time. As in the DSCAM, you have a lot of around 600 possible isoforms. Okay, so at least for one of these splicing sites, it was shown that upon depolarization of the hair cell, the splicing status on this splicing or the specific splicing site changes. Specifically, it is done through a protein kinase that is activated by depolarization, and then the splicing changes, and it gives rise to a different ratio of the slow variants in this cell, which will then would cause it to oscillate in a different frequency. Now, just the last comment here is that HNR and PNSR proteins are known to be excessively modified by phosphorylation. Okay, so again, to conclude the splicing in auditory system, so what we think happened is that initially, well, you are born, and then initially the hair cells are tuned to specific sound waves just because their location on the basal membrane. And then if, for instance, this cell right here will respond to about 60 hertz, then it will cause it to depolarize, changes the slow splicing status, which will cause it to oscillate in different frequency, which will make it more sensitive to the 60 hertz sound wave. So the tuning, and by tuning in auditory and also in visual systems, actually we mean that if we will plot the cell response, neuronal activity, amplitude of response, whatever, but just cell response to different frequency, there will be a single frequency where you will get a peak of response. So this is maybe the picture before you get the fine tuning by splicing, and afterwards this curve will be thinner. So this is a finely tuned hair cell compared to before it was reshaped by splicing. Okay, that's the story of slow in the auditory system. Questions? Okay, there's another. Now we move on to another mechanism that happens still in the nucleus, and we call it editing. So RNA editing involves an insertion or deletion or modification of a specific single nucleotide. the most common editing changes is when you take an adenosine N and changes to a different nucleotide. We didn't mention up till now, but it's I stands for adenosine, I think it's pronounced, adenosine, and C to U. So these are the most common editing we see. And the complex you perform the editing, as in the splicing example, splicing spliceosome, editing editosome, so that's easy to remember. So the editosome is a complex of proteins and which performs the editing, and it also includes an RNA molecule that serves as a guide through which, by which the messenger RNA to be edited will be modified. Okay, we almost don't say anything about more specifically about this complex. But just note that there is this process of editing, meaning changing of a specific single nucleotide along the RNA. So it's not splicing, right? You changes just a single nucleotide. So we call it editing. One example of editing in neuronal context in depression. So it's a very interesting research about 3 years ago that have shown that you take a group of patients that suffered from severe depression, and we talk here about suicide victims. So this is extremely severe depression. It's not, you have to, only in this group you see this modification I'm about to tell you about. Okay, so you take them and compare them with the control healthy individuals, and you see they have increased prevalence or increased frequency of a certain kind of editing mutation, certain kind of editing process that happens to the messenger RNA of the serotonin receptor, serotonin receptor. So 5-HT is serotonin. It's a neurotransmitter that plays an essential role in mood regulation. It was seen in many experiments. And its receptor is being edited differently in severe depression patients. Okay, and then they raised the idea that it might be partially responsible, therefore, for clinical depression. Not sure how well they have proven it in this direction, a causative role of this editing, but there might be more experiments up till now that show this thing. But it's very intriguing to think that an editing of just a switching of a single nucleotide would cause such a dramatic, horrible disease. Yes.
Is it something they have more of the editing site?
Okay, so I think there's a mutation here. I don't say anything about what's the difference in their editing. They have some difference in the editing. What we do know that this difference lowers the efficiency of the response of this receptor to a serotonin. So we have lower serotonin transmission. Effectively, we have lower serotonin transmission. That we do know in these patients, and that might be play some causative role in the disease.
A long time we have compensation.
I guess there is some compensation at the biochemical level that do not cope, with this good enough, I would assume. Okay, so these are all the processes that happen in the nucleus, and we now need to translocate the messenger RNA to the cytosol, where it will be regulated in additional mechanisms. I guess some of them will be we will get to know only last week. Okay, just one comment before we move to the transport across the nuclear envelope. So we mentioned splicing, and we gave some neuronal examples and so on. Okay, splicing is still a mechanism that is thought to happen in the nucleus. That's what most neuroscientists will tell you. But lots of evidence are being accumulated, and some researchers claim to see functional splicing at the synapse site. There are a couple of papers about that. And it's attractive to think of splicing at the synapse site because, well, we know that certain synapses can strengthen upon presynaptic signals, and not other synapses at the same dendritic tree on the same neuron. So that has to be a mechanism that does not pass through the nucleus or through the cell soma, right? It has to be something that's unique for the specific synapse and not for an adjacent synapse on the same dendritic tree. And splicing gives you this possibility, at least hypothetically, because if you have the RNA molecules already at the synapse site, but you don't have some function just because they are not spliced in a certain manner, and then you have some signal from the outside, only in the locally at the region of the synapse, and then these RNA molecules that are already there at the cytosol, at the dendritic terminus, are being changed in such a manner that give rise to different proteins, which give rise to different function, right? And then the synapse, I don't know, it becomes wider or thicker, and the distribution of ion channels changes at the plasma, and so on. So lots of mechanisms that will explain the morphological changes of a single synapse and not all synapses on the same dendritic tree is a topic that lots of researchers are interested in, and some of these mechanisms might be splicing.
Will the translation also occur with synapses?
Translation, yes. You have some ribosomes that sits very, again, some researchers claim you see them, so yes, translation happens maybe not at the same level as around the nucleus, but at a certain level it happens also at the dendrite and the exon terminus. But there's still sort of a debate there, because I think most researchers, I think, will tell you that you see the components, why people think they see splicing in the synapse site, because they see small nuclear RNAs and they see the proteins that are part of the spliceosome right there at the synapse site. But people will say that it might be because they play a part in different mechanisms, which we know is true. But several groups have shown that you put an exon into an exon construct to a cleaved dendritic tree. You just cleave it, just take off the soma, and you can still hold it in a vital manner for a couple of hours, the dendritic tree, and you put an a construct of an exon into an exon, and you see that this construct is being spliced. So they would say that these components are there, and they are functional in the splicing sense. But that's still a debate. Okay, so let's move on to the transport across the nuclear envelope, where we will meet the nuclear POG complex, the NPC. So that's the gate that sits in the nuclear envelope. A couple of words about its morphology. The nucleoplasm, or the inside the nucleus, there's a structure that resembles a basket, and through the transmembranous part, we have the proximal filaments, sometimes called short filaments. There are these filaments. They interact with the messenger RNA while it passes the gate. We will see it in a second. And in the cytoplasmic site, we see the long or the cytoplasmic filaments, these red filaments here. And this is one of these cases in biology where the diagram looks pretty similar to the EM. I'm not sure it's an EM, but to what we see on the microscope. So you see the basket structure from the nucleoplasm side, and that's cool. It's a very huge complex, about 125 million Daltons. That's a lot. Okay, so proteins up to the size of 60 kilodalton can diffuse through the nuclear pore complex in a passive manner. Okay, so this gate allows us to translocate RNA molecules from the nucleus to the cytoplasm, and also translocate proteins that are translated in the cytoplasm back to the nucleus. Let's start with the first direction, because the main topic of this chapter is the RNA. So what we think happened is that this messenger RNA, the splicing reaction that occurs on this messenger RNA leaves in its trace some leftovers, let's call it, of messenger RNP proteins. These are proteins that played a part in the splicing and remained interact kept their binding to this messenger RNA, and then through them you have interaction with several, let's call it adapter proteins, that are the messenger RNA exporter. It's a small complex of a couple of proteins, and this complex in its turn interacts with the short filaments that fill the tunnel of the NPC. And these interactions is the one that enables the messenger RNA to translocate to the cytoplasm. So an RNA has to be spliced, has to be post-splicing to have these leftovers of the splicing proteins to be recognized by the messenger RNA exporter, to be recognized by these filaments, and to be transported to the cytoplasm. Otherwise, it just would not be recognized by the NPC and would not be translocated to the cytoplasm. So that's the reason why pre-messenger RNAs they are localized to the nucleus and cannot pass to the cytoplasm because they have to have these interactions with other proteins. Okay, so as I said, the main rule, the main topic here is RNA, but through the NPC also proteins can pass back to the nucleus. Okay, so we see here that it's a protein that has to, or that passes through the NPC back to the nucleus has to have a specific sequence in it, a short motif that is called the nuclear localization signal, the NLS. And both gain and loss of function of NLS have shown to be consistent with what we think the NLS does. So it's maybe easier to think of a protein that is localized under physiological conditions to the nucleus, and then if you destroy the NLS or take it out from the protein, you would find it at the cytoplasm where it is translated. But it's a more difficult or a more rare case in biology to show what they have shown, that what we see here, that it's there. Okay, so pyruvate kinase is usually a cytoplasmic protein, but it's enough to add artificially, link it with an NLS, with the nuclear localization signal, to cause it to translocate to the nucleus. So indeed, we think that the NLS is the key or the tag that shows the protein the way to the nucleus. And we mentioned a couple of times transcription factors that are being phosphorylated at the cytoplasm and then translocate to the nucleus, where they serve as transcription factors. They bind to DNA and cause translation, right? We've mentioned it in the Candel paper and in other instances. So partly, or at least for some proteins, this change, the phosphorylation changes the conformation so that the NLS becomes exposed, or just it was hidden up till to the phosphorylation step, and then it becomes exposed and is recognized by some proteins, and it is then recognized by the NLS and being translocated to the nucleus, where this protein would serve as a transcription factors. So this is the mechanism. That's the mechanism for some of the transcription factors, neuronal transcription factors, and others we've mentioned in the past. Yes.
How can it become exposed from what was before?
So if you have a protein, and somewhere internally you have the NLS, but it can be recognized by other proteins just because it's hidden somewhere in the middle of the protein. So most researchers would argue, or you have some instances which you see that that's indeed the case, that the phosphorylation changes the conformation of the protein so that the NLS becomes exposed, and then it's recognized by transporter proteins. Proteins that translocate the other proteins to the nucleus, or allow passage through the. Sorry? Translocate. Translocate, yeah. Translocate. Okay, so we've mentioned the processes that happen in the nucleus, and we've managed to pass the gate, and we still have some enemies at the cytoplasm, and the story is not yet over, and we will meet, I think. We'll even start this point next week. Wait a minute, I want to say something before that. So again, just a teaser for next week. We will see that the cytoplasmic mechanisms, and over there also there are some regulatory steps that happen even before translation. Okay, before we move to the protein realm. Okay, so that's it for today. One word, you were asked, and I was asked also to ask you again to answer the teaching evaluation survey. So it is open for a week now, and it is going to be open for another week, I think. So it's very important for us, for everyone, I think, but especially for us teaching assistants. Please answer it. I know you are so busy. Please find the time to answer it. and particularly because in this course we are not too far from the, sorry, from the, I lost my breath, from the critical mass of students that below it, they just don't process the survey. So please, each and everyone, you just go in and answer the survey. It will be very helpful. Thank you. Yes?
It's the only course where the name of the teacher is not correct. I didn't know how to...
It's written, why? It's written Hermona?
And all the others have different options.
It's written Hermona and Shachar?
I only
Two courses.
Two courses?
One of Ramona and one of Shachar.
Really? So you see the same thing?
No, I had it with all the other courses, but this one was the only one where there were no different options. I only saw the
Hermona? Well, it can be if you, I mean, what you say is that you enter the site and then you see, you can answer the course about Hermona, which you obviously won't do because you didn't meet her yet, and you can answer it for Shachar, and then you can only enter for Hermona. It's weird.
Like for the tutorial and for the biology, everywhere I had
Yeah.
options, but this was the only one where
So anyone who has a technical issue, and I would expect an email from you, Enrica, in an hour or so, send me an email, and I would send it right away to the, I would find to who, I'm not sure to who I'm supposed to send it, and I'll try to fix it as fast as possible. Anyone who has a technical issue, please send me an email. Okay, so for next time we don't have anything, no question papers because we didn't finish this chapter yet, and okay, so we'll proceed with it next week. Thank you. See you next week.
לעשות כאילו שהוא לא...